Okay, good morning, everyone. I think we have most of people coming over here now, and I'm just going to do a very brief introduction. And if this is the first time you're joining us on the BioEMTalks, this is our last series today. And so today's program is going to be a bit longer because we're going to have a final discussion in Gather Town. It's a bit for 1 hour. Okay, so right now we are in the Zoom webinar. So most of you, if you are not the speaker, you probably don't have you are automatically muted and your video is turned off. So each of the talk is 30 minutes with 2 innovation talk with just 15 minutes. So there's probably very little time for Q&A. So we ask you to probably hold your question, especially if our time is really tight, to ask during the live chat in the Gather Town when we finish here. Okay, so we are very lucky to have Kurt back to join us and to give his talk that was supposed to be presented earlier. I'm really glad that he can make it this time. And after Kurt, I'm going to give a brief summary of the BioEM Technologies Career Survey that we did along with the Diagnostic and Biomedical Microscopy Focus Interest Group and the Technologies Forum from the MSA. So I briefly did just a 5-minute summary during the MSA and MMM meeting recently, but we really didn't have much to go through. So I will just, and also not many people made it for that session. So I thought I would just present it again to most of the BioEM people here. And these can also serve as some background for our final discussion. And after my talk, we have 2 innovation talks from two vendors or engineers that are going to present their new instrument today. One is from Connectomics and another one is RMC Bockler. And I think both of them have some new instrument to show us. And finally, after we finish here, we will move to BioEM Town to do our group chat in Q&A. And so it will be happening in the BioEM Town Lowick-Quill room. Okay, so during that session, everybody's mic can be turned on and the video is going to be turned on. But we ask people to conserve the burden on the system. If you are not engaging in active chat, please mute yourself and also turn off your video just so we are not overburden the system. Okay, and if you have any question, you use your raise hand emoji in the control panel. So once you raise your hand through the emoji, your name will be automatically moved up in the participants list. Okay, so we will have 30-minute live chat, and after that we'll have 1 hour of open discussion forum. I send you some of the topics we would like to go through, and I invited most of the past speakers coming back to join us as the leader in the field and to give us some of the some remarks first, and then we will open to the floor that anybody can join with a discussion. I would really like to see if we can get some positive actions to help us to promote the BioEM field. Finally, I just want to quick thank everyone who had helped us to get these 4 series completed, and all my organizing committee. And we are all part of the member of Diagnostic and Biomedical Microscopy Focus Interest Group. And it is a small group of people working on mostly biomedical EM. And so if you like what we do here, you like the BioEM talks, we wish you will join us and continue this network and this communication. And I thank Jenny Slopey and Phil Safer that help us to have been volunteering to run as a guide in BioEM Town. Although I see most of people have been through a couple of times and everybody is really comfortable in the BioEM Town now. Finally, I want to thank our vendors who were able to help us and in order to allow us to run these BioEM talks free of charge. And without their help, we would not be able to offer this series. And I also want to thank Carnegie Institute that helped me to host this webinar and helped me with several IT issues. So all the talks are recorded right now, okay? So I'm in the process of getting them edited out, and then I will put them in a YouTube channel, and then the link will be linked back to the BioEMTalks. Again, if you have registration in my file, you will get a notice when the video are available. Okay, so BioEMTalks is always available. Okay, so we have already built it. So we have vendor booths, and the forum has been working very well. And actually, I got some feedback from Christina Mikeva, and she encouraged us, which is also what we were hoping to do, is keep developing BioEMTalks. We would like to put more image galleries and protocol libraries and probably tip and tricks video and reference collections. And I know Bruno and Alice and I will be talking about, each of us have various reference collections, some of them backed up 60 years old, and since these are very precious collections. And so if we can share it within the BioEMTalks, in the BioEM community, it will be really, really good to benefit everyone. So we would also like to have some innovation news. If you have some innovative techniques or you develop something that you would like to share, we wish we can continue to use this BioEMTalks as a communication format for us to keep our network growing, okay? And as I said, the webinar recording is going to also link in BioEMTalks. All right, I'm going to stop right here and pass the mic to Kirk. And I'm going to stop sharing here. Kirk, would you like to introduce yourself? I know most of people know you, but please do introduce yourself and tell us what you have been doing. Thanks.
Okay, thanks, Rucheng, and thank you actually for inviting me to the BioEMTalks and the entire committee. Well, part, I guess, my introduction is that I'm a director of the Advanced Bioimaging Laboratory and a principal investigator at the Donald Danforth Plant Science Center. And if you've never been here, this is what our front door looks like. It's really beautiful. We have a real Missouri prairie sitting outside that we can watch bloom and beautiful colors in the spring and the fall. And my work is primarily focused on plants and the special challenges that go along with preparing them for EM. And so today I'm going to talk about using enhanced heavy metal staining of high pressure frozen free substituted plant specimens for improved high resolution serial block phase EM. One of my challenges is to deal with the difficulties of plants, but then also, a number of years ago, I realized that some of the benefits that you see with these OTO protocols don't always translate to free substituted material because of the solvents used. So I wanted to address that problem with my work. And for those folks who don't work with plants, near the end of the presentation, I'll show you the same technique works quite nice for other biological systems. So let me move to the next slide. Hopefully if I can get that right. There we go. So I thought I would put this presentation in a historical perspective. So I'm going to first talk about the OTO protocol that I now routinely use for conventional protocols for volume EM. And I will tell you, my talk will discuss a fair bit about XRM and why it's helpful, but also these protocols that I'm going to describe will work quite nicely and improve XRM data sets as well, because there's more metal signal and the X-ray can actually get a better contrast, better signal to noise from it. In the middle of my presentation, I'm going to move into, okay, now that we have a conventional OTO protocol in plants, what did I modify to get what I call volume EM of aqueous OTO? And what I mean by that is that, listen, dogma with free substitution is you keep it all in solvent. In order for me to get as much enhanced metallization as I was able to, I actually had to transition them into an aqueous solution and then move it through the aqueous protocols. I just got a better contrasting result. And then finally, at the end of the presentation, I'll show you some other organisms that we did this protocol with, okay? All right, so listen, this is a simplified diagram. If you haven't seen this paper yet by Hua et al., in 2015 in Nature Communications. They actually described a technique for large volume en bloc staining for EM. They were actually looking at whole mouse brains. And they modified the traditional OTO protocols to allow more consistent and uniform labeling throughout large tissues. And thinking that that could also benefit plants, I tried it, and it appears to also benefit that as well. So basically, you do your primary fixation in aldehyde. They do a secondary fixation in osmium. And one step that they do is they go into potassium ferrocyanide, but they don't actually wash out the osmium before they potassium ferrocyanide. So that's one part of it. Then they go and use the thiocarbohydrazide and then go back to osmium. And then they move through the en bloc staining with uranyl acetate and lead acetate, and then infiltration, so forth. So you can read further details. Basically, I followed that protocol closely. Now, again, from a conventional fixation, one of the tools that I use to say, oh, do I have more metal now? And by the way, I'm going to make it clear to you, more metal doesn't necessarily mean everything's better. It just means that you're getting a different kind of labeling that can maybe more be amenable. And depending on the biological question you're asking, it may be suitable. Certainly, a lot of the brain folks get the benefits of this enhanced membrane contrast. Serial block face has the special challenge, particularly when you're using the microtome version. You have the 7 to 8 millimeter working distance that your sample is from the system, the detector, and the lens. And so therefore, you're a little bit more signal starved. And so ways to enhance the metal are helpful. So one of the things that I did was that I would often take like a 350 nanometer thick section and then put it underneath a macro microscope. So this is just a macro microscope image of an unstained section. There's no toluene blue. This helps me kind of assess how much metal is going on. Okay, so I don't have to stain with toluene blue anymore, and I can kind of use it as a tool. At higher magnification, by TEM, I can see my chloroplast membranes are stained extremely strongly, and also the thylakoid membranes inside, which are responsible for photosynthesis in plants, the stacks of Grana, and I can actually see the membrane separation with the technique. Mitochondrial membranes, plasma membranes in ER are a little less pronounced than you'd see in brain samples, but they were still quite good. So we evaluated that protocol with conventional fixed material and 3D volumes. In this case, it's the Thermo Fisher Volumescope and with variable pressure. And we can easily get these three-dimensional volumes. And with this protocol, most of the manufacturers said, wow, these are the easiest plant samples I had to image with. So I would tell you that it seems to work quite nicely. You still have an issue, right? Plants have vacuoles and air spaces, and they're kind of like lung tissue. And so you need to use VP still with these tools. Or with the Zeiss, we use focal charge compensation to get a nice effect. Okay, so you get the idea. We can do nice 3D volumes. And I wanted to give you another reason why I moved into the X-ray realm. Normally when I did either conventional fixation or high pressure freezing and freeze-substitution with osmicated material, I could get images on the left where I could actually see the cells and screen them and be able to, tell where, I'm a fungal plant pathologist in training. And so oftentimes I was trying to look for an infection somewhere inside of the tissue. And on the left, these are resin blocks. These are samples that are flat embedded in resin blocks that I'm imaging. But when I did this enhanced OTO process, the amount of metal in there was significantly increased that I actually couldn't push my light through these anymore. So that kind of moved me into the realm of using X-ray to find my stuff efficiently. So we started, we have a Versa 520 in the lab, and I'm going to mention his name, Keith Duncan. So if you ever do plant X-ray, that he's, I would say, one of the best in the world. We would do 4x low magnification scans of entire leaf discs. Of course, you can cut these in different angles and look at single slices. And then, of course, you can make 3D volumes. So we would take our materials. Normally, I would actually use my microtome and face off so that I have a flat surface. Makes correlation a little bit easier, right? Because if you talk about a round piece of leaf, where do you start to go in and correlate? So I would face them off. And then we would do a high resolution, like a 40x scan in one small subset. And I just want to make a comment. This is a 4X scan. I can get a lot of information about it, but going to 40, we can see a little more. And here's that little same green zone. And so then we mounted the sample for serial block face imaging, and this EM actually correlates exactly to that location. And I have a red box. So we took these at 5 nanometer pixels. We could go down to about 20 nanometer slices, but we'd easily get, you know, 1,000 plus. The only thing that's stopping us is that when you run it over the weekend, you come back on Monday, it could keep on running. And if you look really closely at 5 nanometer pixels, we could see the stacks in the Grana, in the lamella. That spacing is, the spacing between lamellas is, the gap is around 10 nanometers, and a whole lamella is about 20, 22 nanometers between each double membraned component. So we could resolve that quite nicely with focal charge compensation, okay? And then that same purple area, we could then create into 3D volumes. I'll talk a little bit about our use of deep learning to segment. I'm sorry, when some of the early work I used to do, I had my nice little, students who would segment out for us, but I don't have that luxury in a not-for-profit organization. We don't have students that are looking for letters of recommendation to do that. So we've gone into deep learning to help improve the efficiency of segmenting these things out. Okay, and this, by the way, for those who don't know, this is a chloroplast. The red are the Grana, but we also segmented out the starch and the plastic globules, which are components inside. And so now we can use this volumetric information to quantify, okay? If you're interested in seeing how we did the conventional fixation, OTO fixation and correlation, we have right now currently in bioarchives, we have a paper called X-ray microscopy enables multi-scale high resolution 3D imaging of plant cells, tissues, and organs. This is a nice a document to really see the potential of X-ray for these purposes, including non-resin embedded materials. That's only a small part of it, but it's been accepted into our the Plant Physiology special issue of the Plant Cell Atlas Initiative. So you should see that in the next few months. Slightly different than our bioarchives. Of course, reviewers always like to have us do a few more things. So okay. Listen, I was working with X-ray, and listen, the reason I like X-ray is no matter what, my volume in my EM is still only a few tens of microns, maybe if I'm lucky 100 microns. I still want to put things in the context of tissue. So, hey, why not? It's the same protocol in resin with OTO compatible with X-ray. So we actually started playing with, these are cross sections of the mesophyll cells. And you can actually see the nuclei with these hazy things here. And then the little voids inside on the outside of the cells are actually the starch granules. So we did deep learning and trained it to be able to segment those out, and we could get nice 3-dimensional volumes. Of course, it's lower resolution, but then the EM can plug right inside of that, and you can get the context of the rest of the tissue if it's relevant. Now, recently, Zeiss actually has just come out with a new algorithm, which is deep learning based. This is what our raw normal projection would look like that I did this previous image on. And more recently, they now have their Deep Recon Pro that really improves the amount of detail. Of course, it's taking the 3-dimensional volume of the X-ray data and further improving the signal to noise and quality after processing. So I'm kind of excited about this, and it'll improve the kinds and sizes of things that we can see by X-ray, okay? Now, I'm gonna step back for a second and talk a teeny bit about our standard freeze-substitution protocols. And when I was still at Zeiss, the Otegui lab brought a sample to us, and I didn't actually realize it was OTO at the time. And it was of a pollen, an anther, which has the pollen grain making structures of plants. Okay, it's male reproductive structure. And in her protocol, she used 2% osmium tetroxide and acetone, and pure acetone. She used thiocarbohydrazide, acetone mixture, and osmium and acetone, and kept everything in solvent through the whole process. By the way, that's an improvement over regular substitution protocols. The OTO actually does improve the metallization for serial block face imaging. So this is an example of one of the movies. This is published in Plant Endosomes, Chapter 7. In 2020. So if you want to look at that protocol, this is using more classical traditional freeze substitution protocols. I want to note here, if you look at chloroplasts, because I'm going to point this out later, it is, if you look in chloroplast, normally, if you just have solvent-based freeze substitution, the chloroplastic membranes, the thylakoid membranes, do not stain very well. And there's other structures that you may know behave that way. And I want to compare that when we go into the other protocol. What I like about this protocol is it gives you this classical freeze substitution look, okay? I will tell you that as we go into this rehydration phase, it changes how things look, and that's different. We just have to think about how that affects the science that you want to perform. So I modify. Okay, so listen. When COVID came out last year, I was sitting in the lab by myself, and I just had lots of time without customers or very few customers. So I started playing with these recipes, and I spent a lot of time varying the solvent-based method, and I put water in my substitution fluid, and I tried to actually even add a little bit of water into the OTO mixtures and so forth. But if I kept it predominantly solvent, I just didn't get as much metal staining as I'd like. It always would increase, okay? But this new protocol actually made a huge difference, and that's what I'm going to describe. And one of the things that I always do is I add 3% water now. Some of you may use 1%, some may use 5%, or whatever your variation is, but that actually helps improve the membrane contrast in any freeze substitution process, regardless of whether I'm doing it or not. So adding that little bit of water makes a big difference. The other thing that I did is that I actually did the substitution in my normal substitution fluids with water. I would bring them to 0 degrees, okay, which means they're 4 degrees. So I put them in a 4 degrees refrigerator. I'd substitute them up until that point. And then I actually would transition them at 25% steps into finally a 2% aqueous osmium tetroxide. And then I would continue on the rest of the HOA protocol. Okay, very straightforward. So that's basically the secret to get more metal. And we'll talk about later on what other things could be done. Okay. Here's one other thing a challenge I ran into. You know, a lot of plant structures have this waxy cuticle surface on them that make them buoyant when you hydrate them. In substitution, in solvents, they're fine. They sink to the bottom. The second I went through this rehydration step, they floated the top of my substitution fluid. So about 6 months of my time was dealing with inconsistencies in my process, because they would only have exposure to the surface on one side for the chemicals to penetrate. So I reached out to Microscopy Innovations, and particularly Steve Goodman helped me day to day on this. And they had these mPrep capsules, which are kind of cool. So what I did, and by the way, I'm going to give you one more secret. When I run this process, I use the traditional aluminum carriers and the Leica aluminum, baskets. If you cycle them through, the reactions and the solutions turn black. Something is reacting in the aluminum that caused some weird effects in my substitution fluid. And if you've ever used these aluminum holders that Leica has for the free substitution, it actually etched them. So after 2 or 3 cycles of this protocol, they started leaking, and I'd come in on a Monday after letting them substitute, and my substitution fluid had leaked out of the container. Fortunately, I had them in secondary containers. So I stayed away from aluminum. So I went to Steve, and they have these stainless steel critical point drying blocks that actually hold these mPreps. And they have on the bottom of them, they're open. So what I did is, before I figured out that everything was leaking through, I'd still have a couple good runs with them before they leaked. I would actually put my substitution fluid in here, maybe about just up to the base of this. I'd put an mPrep capsule in it, just a single mPrep capsule. I put my substitution fluid in -80, then I would put liquid nitrogen here until it froze. I would drop my high pressure freezing hats down inside of here while it's in liquid nitrogen. And then I would cap these in specimen containers and put, I have a -80. I didn't use the AFS because I just can't fit all of these baskets, and this is not really an AFS compatible. Well, it will work in AFS, but the way that I was doing it. My comment is I would actually bring them to room temperature where the, in these containers, and then I would put the cap in because I didn't want to cause any kind of melting or thermal change. Then you now have 2 of them stacked on top of each other, and they actually act as a seal, and you can move them through the rest of the processing. And by capping that, I now kept my plants completely submerged through all the processing. I'm sorry for that long run, but it's an important detail, okay? Now, we published this in, I think, in Planta a number of years ago. This is a classical freeze substitution of what chloroplasts look like, with classical substitution with no water. This is our conventional fixed OTO substitution using serial block face imaging. And here is the aqueous OTO protocol. So we're seeing very, very, very strong contrast of these membranes, which, if you remember, when you don't have water, they look like this. When you run through this protocol, you suddenly get this increased membranes here, which is helpful. I'm not saying every membrane is going to do that. Here's an example of using just solvent OTO in the membranes, and then here's the aqueous OTO. So again, you can see the difference. I always got an increase of even doing solvent, but going into this aqueous, I got more. Okay. I'm actually running out of time a little bit, so I'm going to move a little bit quicker through this. We actually, and I'm hopefully you can give me a few minutes, Ruching, to get through this. But so we actually would take our X-ray micrographs. Of course, they're 3D volumes. You can reslice them. Then we would go in and do serial block face. So this is actually the block face using the Thermo Fisher Volumescope. And you can see the EM image at low resolution. And then we would go to high resolution using correlative. And then, of course, we would make 3D volumes. So again, the X-ray also helps me to find if there's any damage in the tissue before I go in and section it as well. They're black. You can't see them through the eyepieces. You don't always know where the damage is. Here I can actually know the ones that I'm going to go after are in good shape. This is just showing you another anther that I went after. So these are anthers. The pollen spores are made in the middle. The tapetum is a very metabolically active layer that provides nutrients. This is a 350 nanometer unstained thick section that I use for guidance. And then here is what a focal charge compensation looks like. Oops, it jumped ahead a teeny bit on me. Of the serial block face of the anther, you can see very, very strong contrast in these samples. If you look closely at these, you'll also notice that the chloroplasts now have dark staining thylakoid membranes. And at 5 nanometer pixels with focal charge compensation, I can resolve the spacing easily between the grana. Okay, so that's telling me a little bit about the resolution. And remember, with focal charge compensation and serial block face imaging in particular, you know, the amount of metal does influence whether you can see the stuff that you want to. So when I'm talking about resolution, I'm talking about the ability to give enough metal to get a good quality result. So I then actually, this is going to be kind of cool. We did this in roots. So we just kind of moved through the different plant structures. Here's the serial block face of the entire root cross section. And here's the 3D volume through the tissue, slicing through the tissue. We were able to take 10,000 by 10,000 pixel images at 50 nanometers step size through thousands, well, I would say 1,000 plus and minus slices very easily. And here's the resulting volume. And then a little bit higher zoom, we can actually see the structures a little closer. And then finally, we can even see, if you look, you'll see the plasmidesmata, the nuclei, the mitochondria, the cisternae and the cell walls labeled quite strongly. And if you look at this, I want to point out, this is one line average. You know, a lot of times if you do some of the work on the serial block face with a 2 microsecond dwell, you have to sit there longer to get good enough signal to noise. This wasn't an issue, okay, because there's just that much more metal in it. The other thing that was kind of cool is the cell wall and Golgi stained quite nicely. So the cell wall, you could easily see the plasmidesmata. If you look at it in where normal traditional stains, normally the cell wall is more transparent and it's hard to distinguish those two. It was very easy for us to see plasmidesmata, ER, mitochondria, and nuclei, okay? We've been doing a fair bit of deep learning now, and I've been satisfied with the ORS Dragonfly. With this technique, I generally have to use a larger patch size, but this is actually a training of a few slices, 8 slices, and we can see the mitochondria, the Golgi. I didn't clean this up. This is just kind of raw. I did a pretty decent job, so now cleanup should be much quicker. And this is what a 3D rendering of the cisternae, cisternal vesicles, mitochondria, and look at the pit field now in 3D. You can get a whole different impression of, you know, the extent of plasmidesmata when you actually put it all together. I make one more comment because I'm almost done. I need about 5 minutes more. I published some work actually with the Delaware lab, and I see a number of my colleagues, Gene Ross, and it looks like Shannon Modler here that helped us do this. where we did a 3 nanometer resolution reconstructions in yeast using high pressure frozen freeze substituted yeast. And at that protocol at the time, we kept it all solvent based. And I'm sorry, I took this as a screenshot. I don't have these figures anymore, the raw figures. I took it out of our biotechniques paper. This is what the TEM image looks like. This is what the yeast looks like in FibSem. The same exact block. We just sectioned it and it has a little different appearance. And you could see, you know, the ER and so forth on it. We actually use 1.5 kV, 3 nanometer pixels, and an average of 30, just to kind of give you a sense back then of how it was done. Now with this QFS technique, I actually worked with Kedar Narayan's lab, and I said, hey, Kedar, can you reproduce this on some other organisms at the NCI? And he actually took yeast and he used it. He used the quick freeze substitution. So the same exact protocol, basically, he now uses the quick freeze substitution. So it works as well when you're doing fast freeze substitution method. This is what a normal AFS solvent UA yeast would look like under the same exact imaging conditions. This is what the yeast looks like, okay? So I zoomed in a little bit closer. This is a STEM image, so we can control the quality. They're 10 nanometer pixels, 600 picoamps, 1 line average. By the way, I'm not trying to cheat here. You can get a better image with the AFS. You have to integrate longer, increase the gain more, and so forth. So you can kind of get the idea of the throughput. This is higher mag, just to show what the structures look like. We can see the spacing in the nuclear envelope. There is a granular appearance to this technique. I think it can be improved, but depending on what you're going after, certainly some of these major organelles. The other thing that you'll notice is that the glycogen particles, which are transparent, normal freeze substitution, are heavily stained with the OTO protocol. So that, again, it's carbohydrates, the cell walls, structures that seem to have that heavily stained. This is a comparison of the AFS protocol, what the yeast looks like. This is how much extra contrast you're getting. So again, depending on your question, you can't even see the nuclear envelope. This is the nuclear envelope, believe it or not, with this particular protocol, okay? All right, last organism, and then I'm closing up here. He also did this in C. elegans. C. elegans, you basically have to do freeze substitution to get a decent result under identical conditions using our OTO protocol. These are 10 nanometer pixels over a large area. And then here's the same exact STEM imaging conditions, 1 average of his nematode. And then since they're such high resolution, we can zoom in a little bit closer and see that we can see the nuclear envelope, ER, membranes, again, 10 nanometer pixels. You get much higher contrast, much faster volumes, depending on your question. All right, my summary. Full rehydration of freeze substitute samples followed by aqueous OTO and end block staining significantly improves sample staining and conductivity for serial block face and FibSem. Protocol appears to increase metallization of all cell structures, but selectively enhances carbohydrate-dense cell structures. Mitochondrial ER and nuclear envelope are modestly improved. They're still there. I wished it was better. This approach is compatible with quick freeze substitution, the AFS, and manual freeze substitution protocols. XRM is highly beneficial looking at these dense structures for multi-scale imaging. It's broadly applicable to diverse organisms and difficult to fix organisms. But listen, there's so many permutations. You can change a lot in this recipe on what substitution fluid you're using. I think there's a lot of opportunity here. Right today is where we are now. I wouldn't do structural biology studies of it, but I think there's a lot of things that we can do and improve it. And I had a lot of help. There's folks in the Danforth Center, the Kedar Narayan's lab, Marissa Otegui's lab to help actually do the initial work that we did on her substitution protocol. Microscopy innovation, specifically Stephen Goodman was with us day to day on Thermo and Zeiss for helping collect some of these data volumes. Okay, I'm done. Sorry for being a few minutes over.
You don't have to rush.
Okay, I talk fast as it is, but I was a little bit. Oh, one more thing, just for the group. Probably within 1 month, we'll have this submitted with a full detailed protocol where you can get all the nuances and, solution A, solution B, and then we'll publish it immediately in bioarchives. And if anybody's interested, they just fire me an email and I'll let you know when it happens.